Okay. Is this too dark in here right now? Okay, all right. So we agreed to do the exam review today, right? Okay. So if we're going to do that today, um, so spend some time at the front end here talking about what you need to know for tomorrow's lab. So I'm going to be jumping between the gymnosperm PowerPoint and the angiosperm. They both should be posted, but you're going to be looking at both of those in lab tomorrow. So I just want to make sure that you've heard it once here and then you'll hear it again in a slightly different way and in more detail in lab. And then Wednesday, I'll come back and probably continue on with the gymnosperms and then switch to the angiosperms. Okay. So with the gymnosperms, um, with this group, I'm not going to be asking you to know their life cycle in any detailed way. However, with the angiosperms, that will be something that you'll learn in more detail. But there's a few things about the gymnosperms that I'll focus on, such as the different types of cones and their function, and some of the basics of what they do in order to make more of themselves. So this slide is just trying to compare the difference between gymnosperms and angiosperms. And then, they're both groups are seed plants. They produce seeds that are the unit that they disperse out. Whereas the other groups, the bryophytes and the seedless vascular plants, they release spores. So it's a very different strategy. And when you look at the seeds and some of their characteristics, they say that it really does improve their survival compared to just the spores that were released in the two other groups of plants that we looked at. The seeds are capable of surviving for longer stretches, especially in more challenging environments. And so this was a big step that made these two groups of plants pretty successful. And the angiosperms are the most successful. If you look at the sheer numbers on the face of the earth, they win. And with the gymnosperms, gymnos means naked and sperma means seed. So naked seeds. And that's how they actually refer to them. And that's just getting at the point that they're not buried in any kind of fruit tissue. They're sitting right on top of the scale of the cone. When you look down on a pine cone that has its scales open, you might be able to see those seeds sitting right there. So the pollination step, so if you look up the definition of pollination in a dictionary, they almost always will give you the angiosperm definition of how angiosperms do it. This is how gymnosperms do it. So pollination is when the pollen goes directly to the ovule. If you look up the definition in a dictionary for pollination and angiosperms, they'll say pollen transferred to the stigma of a flower. And that's true. It's just angiosperms, but you have to also think about the gymnosperms. And they don't have flowers, so they don't have stigmas as part of the flower. So they kind of have a simpler way of doing this. They just take the pollen and get it right to the ovule. And I'll show you pictures of this. So gymnosperms are what some people say they're fruitless seed plants. That's another kind of heavy on the angiosperm idea, where the gymnos are lacking in fruit, but they do have cones, and angiosperms don't have that. So another big jump is that they don't need water anymore to get the sperm to the egg. So again, that's a good strategy. It's very successful compared to the other two groups where we said free water was necessary. So you always had to have free water, and that's a limiting factor. With the gymnos and the angiosperms, we don't need to see water in the environment anywhere. It doesn't have to happen. And so again, this is an advantage for them. Now, there's a couple of terms you're going to start hearing now that will come into the angiosperms as well. We say that seed plants are heterosporous. So we're going to find that they do make spores, they just don't release them. Okay, so they have a slightly different strategy. And so we're going to find out that there are two types of spores that both the gymnos and angios have. One are the megaspores, and then the other are the microspores. Anytime you think about mega, think about female, all right? Something to do with the female. And so the way I remember that is mega has EG right in the word. You add one more G and you got an egg, okay? And females have eggs. So megaspore will eventually develop into megagametophytes. And again, that's the female. Some people would say female gametophyte. It's the same thing. Megagametophyte, female gametophyte. Microspores, I always equate that with sperm are way smaller. They're tiny, tiny, tiny in comparison to eggs, so they're micro. So anytime you see microspores in botany, it's something to do with males, okay? And so microspore will eventually develop into microgametophytes, and that's the male gametophyte. Okay, so I will show you a little bit more about this with some pictures to have it make a little bit more sense. But we'll touch on some of the big picture ideas. I do want you to memorize these four phyla. That's the only four. That's it. Okay. So phylum Pinophyta or Pinophyta are the conifers. So when I say the word gymnosperm, that's probably what you're thinking. You're probably thinking about, white pine, red pine, balsam fir, and on and on and on. So they're the biggest group. They're the most successful in terms of sheer numbers. These phylum Cycadophyta are the cycads. And some people, if you look at them, you would say that's a gymnosperm because they don't look like gymnosperms, but they are. And Ginkgophyta is another one. You'd go, that's really a gymnosperm? It doesn't look at all like a gymnosperm, but it is. So Ginkgophyta, and then Nidophyta, the G is silent on that last name there, Nidophyta. There's only three genera that are still living. All the others have gone extinct. So we will primarily focus on the first one, Pinophyta or Pinophyta, because that's the most abundant in terms of numbers and species diversity. But I will briefly mention those other three. In lab, you probably won't focus very much on those. You might look at some Ginkgophyta leaves because they're very unusual for a gymnosperm. But in lab, you'll focus on the Pinophyta. So we've had a brief introduction to this idea, just kind of touched on it in lab a couple weeks ago, I think. And so we haven't really got into it until I'm just going to mention it a little bit here. And then when we get into the angiosperms, we'll really dive into it more. But here are a few key things to think about. So when you look at how plants go through their reproductive life cycle, they have like two phases. There's a haploid phase and a diploid phase. So most of your body is diploid. So most of your cells, you have 23 chromosomes from your mom, 23 from your dad. And it's only your sex cells, the egg or sperm, that are in the haploid state. Okay, plants are a little different depending on what group you look at, but they still have this shift from haploid to diploid. So when you go out and you're hiking around and you see a beautiful conifer tree, what you're seeing is the sporophyte phase. All right. So we say that the gymnosperm, the dominant phase of a gymnosperm is the sporophyte phase. That's what you see when you're out hiking around and you see all these beautiful trees, the conifers. It's the sporophyte. You won't even see the gametophytes really until they're you'll see pollen grains eventually, and that's the male gametophyte. But the female gametophyte you won't even really see. You'll never see it unless you dissect a cone. Okay, so it's the sporophyte phase that dominates. It's the same with the angiosperm. We're going to find the same thing. So most of their tissue, when you look at the tree body, it's going to be in the diploid state. That means it has the full complement of chromosomes. So if they're going to need to make sperm and egg, all right, they have to have that happen to complete their life cycle. So if you want to go from a diploid state and make sperm and egg, which are haploid, you have to go through meiosis. All right, so meiosis is the process that you probably learned about in BI 111, where you essentially cut the chromosomes in half. All right, so then you go from diploid to haploid, or another way to write it is 2N to 1N, and sometimes they just drop the number 1 off and just say N, 2N to N. Okay, so diploid to haploid. In order to do that, you have to go through meiosis. And what you see here is that when you look at a sporophyte in any plant, any of the sporophyte phase, their job is to produce spores. So it's right in the word. Sporophytes produce spores. Sporophyte produce spores. That's their job. So that's what you see happening here. By the time they go through meiosis, their spores are mature once they complete meiosis. And then these spores will start to grow and develop just by cell division. And that's just plain ordinary mitosis. Okay, so that's just cloning the cell, making, division, cell division. So when the spores start to grow and develop, they develop into gametophytes. That's the other big phase, okay? So the gametophyte is the other, the alternative form of the generation, right? And the job of the gametophyte phase is to make gametes. So it's right in the word. Gametophytes make gametes. Gametes are sperm and egg. So it's in the gametophyte tissue that you would zoom into if you want to find sperm and egg, all right? And then once you get the sperm and egg fully mature, ready to go, then hopefully you'll get fertilization where the sperm and egg will fuse, and that is called fertilization. If you do that, then you're back into the diploid state. Once the sperm and egg fuse and fertilization has occurred, we call that resulting cell the zygote. So that's the initial stage of the diploid sporophyte phase. And then that zygote has to grow and grow and grow and grow to become the sporophyte, whether it be a white pine tree or a giant redwood. All right. Now, when you're studying this, I highly encourage you to draw this out over and over until you're comfortable with this whole idea of how to go from sporophyte to gametophyte, how to go from gametophyte to sporophyte, which one's diploid, which one's haploid. Okay. So I will be asking about this on the next exam, not this exam, but the next one. So I think it would really, really help if you draw it out, maybe talk it out as you're drawing it so that you feel comfortable with this, because it's not going away. You're going to get it in even more detail in the angiosperm. All right, let me show you a couple of things that I think you've seen this already. So one of the big points from this slide is that you see the bryophytes are releasing spores, okay, not seeds. They're releasing their spores out into the environment, and they're kind of like, you're on your own. If you hit a good spot and you can start to grow and divide and grow into the gametophyte, great. All right, so they release their spores. The spores develop into the gametophytes. Okay, we just saw that on the previous slide. Spores develop into the gametophytes. So with the bryophytes, there you see a moss example. With a seedless vascular plant, it's actually when you're out hiking around and you see those beautiful ferns, it's actually the sporophyte phase that you're looking at, not the gametophyte. And again, though, you see the spores are released. They release them out into the environment and they're like, you're on your own. Hope you can make it. All right. So when you compare that to the gymnosperms and the angiosperms, that doesn't happen. The spores don't get released from the sporophyte. They are retained. And that is a big bonus. That's a good thing. It seems to have helped their survival. And so here, number one, it says spores remain on the sporophyte. And then they're going to grow and develop into gametophytes, but they're all retained in nicely protective tissue. So they're kept well protected as they develop into the gametophyte and make their sperm and they make their egg. Right? And then if that all goes okay, then the sperm and egg will fuse and you'll get the development of the zygote, which then develops into the embryo. which is like the little baby plant in the seed. Right? So when you crack open the seed, there's an embryo in there, and hopefully some good nutrition that the embryo can utilize when it's first germinating. All right, so that's a big shift, and that's important to think about. All right. Now I'm going to jump ahead because we don't have time to get into more of that because I want to show you some things you're going to look at in lab. So in lab, we're going to find some images of the pine cone, right? And so when you look at conifer cones, pine cones, there's like the male version and the female version, okay? And so here you're looking at a female. Ovulate means female. And so the ovule is just kind of sitting on top of the scales. If the scales are open and you look down on them, the ovule's there. It's hard to see with the naked eye, but they're there. And that is where you're going to get an egg eventually to show up. And the pollen has to do its job of getting the pollen grain there. So I'm not going to go through this in detail, but you do see this idea of meiosis occurring, all right, to get into the haploid state. And then you can see fertilization down here in the lower right. That's when the pollen will have gotten over to the female cone and the egg and sperm can unite, confuse. All right. And then if everything goes okay, you'll get the embryo, which is the baby. It's like the baby sporophyte sitting in the seed there. Okay. All protected in that seed. The pollen that some of you may be allergic to, you have to look for the pollen cones. And we're going to see how the microspores will go through meiosis and every single one can develop into a pollen grain. So there's lots of pollen with the gymnosperms. They produce a lot of pollen. Those of you who are allergic can appreciate that. And so their job is to get the pollen to the female cone right where the ovule is sitting on the top of the scale of the pine cone. So you're going to look at a variety of different cones. A lot of them are the female cones because they persist longer than the male cones. They're easier to have on hand. So here's some interesting pine cones. You hopefully can see some cycads. These are cycads, and they have really big pine cones. And then this upper image here, that is juniper. They have tiny little pine cones. Those are cones, but people might say, oh, look at the juniper berries. You might have heard them talked about that way. Juniper berries. Do you know what we get from juniper berries? That's probably $1 billion business. Cough syrup, yes. Something else even more lucrative. Gin, the alcohol, gin. So that is actually coming from juniper berries, but that is actually a cone, not a berry. So it's kind of odd, but true. So these are some kind of unusual examples of gymnosperm cones. These are more like what you're used to seeing. And so here on the left are some pollen cones. You can see they're smaller than the female cone of the same tree. And here you can see how the scales are open. And so they tend to open up their scales when they're waiting for pollen to get there so that the pollen can get in there easily enough and land on the ovule of the cone. I'm not going to go into the words here. I'm just going to show you pictures for now. Here is, if you're allergic, you probably don't like that image, but you can see how much pollen is being released by the male cone. And then if you look down on the female cone, the seeds, if everything goes okay, there'll be 2 seeds on each scale of the cone. Did you have a question?
Yeah. Did they drop the cones before
They'll drop the cones when everything's done, like done. The seeds are gone, then they'll drop. Oh. Yep. And so I'll show you some pictures of the different stages of seed cone development. So when you look at pollen cones, you can see they're way smaller and they're a little bit simpler. They don't get woody like what you see with female cones. This is a male cone under the microscope. So you'll see this in lab. And all those little dots are going to develop into pollen grains. Okay, so lots of pollen grains. Here you can see on the left cones That's all the same tree, and you can see the cones are in different stages of development. So some, like the one in the foreground, is pretty wide open. So it's either just released its seeds or it's waiting to be pollinated, one or the other. Whereas this one on the left looks like it's partially open. It's probably getting ready to be pollinated. This one's all closed up. So the scales will fuse together after the pollen has got there. Then they close up. They're like, okay, we're protecting this because it takes a long time. Once the pollen gets to the cone, to the ovule, the scales grow together, and then it will stay like that for over a year. And then eventually it'll open up when the seeds are ready to be released. So they're very protective of everything going on in there. There's a lot of stuff going on. They just protect it by closing up. Here's another picture showing different growth stages of the cones. So the newest cone's usually at the tip of the branch. And then a 1-year-old pine cone, if everything went okay, there's a lot of stuff going on in that cone. It's just all closed up to protect everything going on with seed production. And then this one is wide open. I don't really, oh, it looks like there might be some seeds in there, actually. So there's some seeds waiting to get put, sent out usually through wind action. So the female cone, you'll look at some examples in lab. So if you look at that spot, it kind of looks like there's something going on right there where my laser pointer is. That's an ovule. And there's 2 of them. You just don't see the other one. It's behind that one. Right? So the ovules are sitting right on top of the scales. There's 2 of them per scale. So that's a scale right there. And there's an ovule, there's an ovule, there's an ovule. So that's the female cone. Here's a blow up showing the ovules. Okay, here's ovule tissue here and here. So the female cone's a little bit more complex. And so that's an ovule right there. Okay. So the pollen has to get there. And if everything goes okay, then eventually there'll be seeds. You'll also look at pine needles under the microscope, and they're, we say pine needles. It's okay if you say leaves, although people will actually say that true leaves are actually leaves of vegetable. A lot of plant scientists say that the needles are actually short branches. But I'm okay if you just say leaves or needles, say pine needles. And so you'll look at them when you have a baby seedling, they just have single needles. They don't group them in any way. But once they get a little bit older, they'll start shifting their growth pattern to have different groupings. And so that's how one way you can identify different conifers, like red pine has 2 needles in a fascicle, a bundle or fascicle. White pine has 5 needles in a fascicle. All right, so it just depends on the plant. And so you'll look at some of these under the microscope, and we'll be asking you to find the xylem and phloem and the sunken stomata or recessed stomata. So that's what you'll see in lab. Okay, so you'll be looking at some cones, some needles, and a little bit about seed development in the gymnosperms. Okay, I don't have a lot of time. I wanted to say a couple of things about angiosperms before we break for the exam review. So I'm going to shift over. So with the angiosperms, couple of things. You're going to get more into the life cycle. One of the things you're going to do is look at flower structure. So we have some lilies for you to look at tomorrow in lab. And you should be able to identify the different parts of the flower. So on the exams too, we'll be asking you flower parts, okay? The only thing I don't really care too much about on here is like the word peduncle. I just say flower stalk. So peduncle, you don't really need to know. I don't expect you to know perianth either. I don't really say corolla. I usually just say petals, sepal. I do want you to know receptacle though. That's the very base of where the flower sits. on the stalk. And I need you to know that because that sometimes plays a role in fruit development. So please know receptacle. And then all this other stuff we'll get into. So you'll work through this in lab and identify with some lily flowers. And then, so when we look at the flowers, we're going to zoom into the ovary, which is the swollen base here. It's usually right in the middle of the flower. And then when you dissect into the ovary, you might have one ovule. You might hear there's 6 ovules. It depends on the plant, what you're going to find. And then if you zoom into the ovules, that's where you find the development. And I'm going to go through this in more detail in lecture, but basically in the ovules, you have this process where you end up with egg formation and then some other cells that are involved in getting everything to happen correctly. So we're going to work through this. It's fairly straightforward. As you can see, it's a pretty short process here. But once you get that egg in the flower, in the ovule inside the ovary, the egg gets formed, and then it has to wait for the pollen to land on the flower. So when we look at that, we're looking at this idea of, where did it go? We're looking at the idea of pollination, where the pollen grains are landing on the stigma. So that's the definition of pollination in angiosperms. Then the pollen lands there and they start to grow out this long pollen tube. And then the sperm will go into the tube and migrate all the way down to get to the egg. And then once that happens, the sperm and egg fuse and you start getting the zygote and the embryo. And it's the baby sporophyte starting to develop inside the seed. And then that flower, parts of the ovary and sometimes other parts of the flower will develop into fruit tissue. So you get this huge shift from this beautiful flower that then parts of the flower has to morph into. I always wondered about that, like with apple blossoms, when you look at apple blossoms, they're so beautiful and fragile. Then you end up with this really tough, hard apple. And that's, flower tissue that morphed into and developed into the fruit that then encases the seeds. So really interesting development. So we'll go through that in a little bit more detail. You will look at some slides in lab showing the general process of how the, like you'll see some slides under the microscope of this process, but we're not going to you don't have to know each step visually, because it's way too detailed. But I'll go through this with you in lecture and I'll tell you exactly what I expect you to know for this, okay? But you'll look at, it's just kind of neat to look at what's happening in a real ovary. So this is ovary tissue and here's an ovule. And you have some of those cells that I just showed you in that cartoon drawing that are showing up. All right, so just looking through this and getting a feel for what does an ovary look like? What does an ovule look like under the microscope? And you can see that's enclosed by integument tissue, we call it, very protective, trying to keep everything well protected. So you look at that under the microscope, and then you'll look at some flowers. Then you'll also look at pollen. Pollen grains are cool because they are all kinds of different designs. And of course, they have a very important job because eventually sperm shows up in the pollen grain, and you've got to get that sperm to the egg base of the flower in the ovule. And you'll look at some cool looking pollen grains under the microscope. They vary depending on, this is what we call an opening or an aperture. In monocot type angiosperms, they just have one aperture of their pollen. So you can tell under the microscope at least, hey, that's a monocot because it only has one opening, one aperture. This one has 3. You can see kind of 3 slits or openings. So this is a dicot. So you can just instantly tell that this is a dicot pollen grain. And you can see how different they look from each other. And so it's fun to look at them under the microscope and get a feel for, they're so unique. And they can be used, they've been used in forensic anthropology, I guess, to solve murder cases because they're so specific. that they have been able to track down murder suspects based on the pollen left at the crime scene. So, okay, so you'll look at pollen grains as well under the microscope in lab. All right, I'm going to stop there. And so I wanted to spend a little bit of time going over the content for the exam, which is Friday. Let me post. Resume recording. Okay, so I'm going to do this. Okay, so now you can see all those slides from that plant and cells. So a lot of terminology. Not my fault. They just, any field of study you go into, there's going to be terminology and jargon that you need to know. So it's no different in the sciences. So yes, sometimes the words are long and annoyingly hard to pronounce. Do the best you can. I like to look up the meaning of the word to help me learn these better. So we got into parts of the plant cell. We got into cell wall structure. So I do want you to know the cell wall, what makes up the cell wall in general. We talked about the cellulose and, you know, the fact that that's just a bunch of glucose molecules all put together into those microfibrils. So we don't have cell walls, plants do. And not only that, but some parts of their body actually have two cell walls. All right, so that's very different than the animal body. So I want you to be able to think about that, as well as some of the unique things with the cells, right? So we have that huge organ vacuole, right? Chloroplasts, unfortunately, we don't have those, so, but plants do. So for sure, think about some of the unique things that plants have. Are there questions on any of the content in here? Yes. Yes. So the leucoplast is like the umbrella term, right? And then the amyloplast or starch, so you'll have like the potato tuber, and then the oleoplast or all the fats and oils like the avocado. Yeah, you pretty much, I mean, you can kind of see them with the oleoplast. I don't think you stain those in lab. They're visible. It's a little challenging. Amyloplast we usually stain with the iodine. Whoops, the iodine. Other questions? Yep. No, no. Know the cell anatomy, including the cell wall. Then we got into plant tissues. So thinking about the different types of plant tissues, including the meristematic regions of the plant body. Questions on the plant tissues? Yes. Yes. Xylem and phloem are complex tissue. That just means that there's more than one cell type that make up that tissue. And so with the xylem, we have two subtypes of xylem, the vessels and the tracheids. So here you're looking at vessels, and vessels are wider in diameter than the tracheids. Here's the tracheids. They're skinny. And rays are part of the xylem tissue too. That's just getting, like, if you have a huge tree trunk, The rays help deliver the nutrients out across the whole tree trunk. But I'm mainly interested in, when you look at xylem, it's tracheids or vessels. Those are the two big comparisons to think about. And they're dead at maturity, which means they have a really thick secondary cell wall, right? Anything with a secondary cell wall is dead by the time it's mature. And they consider the vessels more efficient. And that's why they think one reason why angiosperms are so successful, because angiosperms have vessels, but gymnosperms do not, right? And so angiosperms can have both vessels and tracheids, whereas gymnosperms are limited. They just have tracheids. So knowing xylem has like tracheids and vessels. And then with phloem, we're looking at the sieve tube members or sieve tube elements, same thing. And then they have a neighboring companion cell. It's a one-to-one relationship. For every sieve tube member, there's a companion cell right next to it to help out. And so that's in the angiosperms. We know a lot more about sieve tube members than we do about the sieve cells, which are found in gymnosperms and ferns. The seedless vascular plants have sieve cells. And we don't know a whole lot about them, but that's the other type. And so also remember, phloem are not dead at maturity. They still have cytoplasm, they still have some organelles. So they're pretty different compared to the xylem. Other questions?
So
No. Nope. Yep, they just have one or the other when it comes to phloem. Its own category. So meristematic, kind of think of that as a very unique, special category of active cell division. And so they're kind of their own little beast. And then ground tissue is like what makes up the bulk of the inside of the plant body. And that's mostly parenchyma. If you were to count up like what's inside the plant body, most of it's parenchyma cells. Other questions on any of the, yes?
we have to be so, we have to point out.
Might have to label. Yeah, if I were to do something like that, I either show you a picture and then ask you a multiple choice question about it, or I might have a picture with a word bank and then you label it. So there might be a labeling picture where you might need to, but the word bank would be there. So you would just do like that drag and drop idea. Yeah. Other questions on this slide set? This was probably the heaviest slide set in terms of terminology. Lots of terminology. I feel your pain. I remember what it was like. It's a lot of really long words that are hard to pronounce. Okay, then we got into the plant body, which is basically looking at the root stems and leaves. I separated it out into three different, this is my version. I have it all in one PowerPoint slide set in my hard drive, but I have it as root stems and leaves and EduCat. So root anatomy, the basics of root anatomy overall with the taproot versus fibrous, but then also looking at the zones. So slide 5, when you look at the different zones of the root tip area, you know, just the tip leading all the way back to the zone of maturation, the region of maturation. The slide 6 here, where we look at what's happening once water comes into the root. So thinking about that idea and how we have that endodermis, that ring of cells that kind of regulates water flow into the xylem. So the terminology on here is important, looking at what what is an endodermis with the Casparian strip, which is that orange waxy stuff. That's wax, really thick wax. And basically the endodermis just nicely regulates water flow, so it's forced to go one way all the way into the xylem and not backflow out of the root. What else? Comparing monocot and dicot with the angiosperms, looking at the different design of root tissue. So when you look at the root tissue, you're really trying to zero in on the vascular tissue, the xylem and phloem, and how it's designed. And it really does depend what plant it is. It's a monocot versus dicot, angiosperm versus a gymnosperm. It's a little different. And then don't forget to think about some of the specialized roots idea. I gave some examples of specialized roots. That might be something where I just ask it without a word bank, like, tell me something. All right, so think about that. Specialized roots, do you mean specialized roots itself or examples of plants that you don't necessarily have to know the plant itself, but like crop roots, like that's specialized, like what's that good for? You know, so more the function of it, not like which exact plant has it. More like what is it? What's its function? Why is it special? So same with the stems and leaves. If there's anything special that I mentioned, you know, and there's some examples. So stem tissue again, we're looking at monocot versus dicot stem tissue design with the vascular tissue, how that's arranged. That's a pretty important concept. So please think about both with the root and the stem, what we got going on with the design of vascular tissue. Yeah, so if you see the word mesophyll, like they do that in the leaf tissue a lot. Where is that? Yeah, so here. And that's they don't stay consistent, unfortunately. So you see here palisade mesophyll. So mesophyll and parenchyma mean the exact same thing. I try to say parenchyma because that's what it is. I don't know why they came up with the word mesophyll. It's yeah, I guess, right? Yeah. So yeah, it's annoying though how many times they can use a couple of different words for the same exact thing. Whoops. What happened there? Did I close all the way out of it? Looks like it. Didn't mean to do that. Okay, roots, stems. Stems have to breathe, so they need those lenticels for breathing. Specialized stems, so rhizomes. So this slide 25, like the terminology here. And that's important. Specialized stems, huge stems. And then we got into leaves, leaf design. So both the surface of the leaf as well as what's happening internal with the anatomy. That's important. And then we looked at the difference between simple and compound. Do you remember how to tell the difference between that? What are you looking for? If you're looking for, is that a simple leaf or more complicated compound leaf? Whoops, that's not what I wanted. The axillary bud. Yep, where did it go? So if it has an axillary bud at the base of the petiole, so here's the leaf blade, the petiole, to that. So that is here. Here, you don't see any axillary bud. Base of the smaller. You only see it. Then you'd say that entire thing is a leaf and it's composed of leaflets. So that right leaf has 5 leaflets to make up the leaf. Vein pattern, monocot versus dicot, that's a big thing to think about. I will not ask you slide 45 where it's looking at all the leaf morphology, diversity, and all these. I will not be asking that. Just know it's highly diverse. When you look at this part of the plant body, it's the most diverse part of the plant. Questions on anything in this slide set? Okay. We're running out of time here. We got into the fungi and algae. So we didn't really get into heavy detail on all the different phyla. There's only a few in here that you need to know. So the classes of the fungi, please recognize those words. So slide 8 here, where it shows the zygomycetes, ascomycetes, be able to recognize those. And then the different, you have the hyphal growth form or the unicellular growth form. And then we talked about mycorrhizal fungi a little bit. Algae, there's a few phyla that I want you to know. So the green algae, oh, first let me go there. Morphology, again, the body of algae, how they can be single-celled or all the way to kelps. And then we only really covered green algae, red algae, and diatoms, right? That's it. Mm-hmm. Yeah. So the chlorophyta, rhodophyta, and chromophyta. So the phylum names you do need to memorize. Those will show up. And then with the bryophytes, we did a lot of it is, that there's 3 different phyla in there too, and we were looking at who belongs into each of the phyla. So knowing that you have the bryophytes are the mosses, and the hepatophytes are the liverworts. All right, so though matching what organisms belong into each phyla, the basic kind of morphology of these 3 groups. And there's 3 different. So mosses, liverworts, and hornworts. Questions on any of that, or the fungi or algae? Yeah, so we are listing out like applied uses or interesting aspects, so be able to either recognize or state something. Doesn't have to be super detailed. Like, I'm not asking you to spit back in any detail, but knowing that they're going to look at the drug discovery, secondary metabolite, that type of idea. What about with the seedless vascular plants? Again, the phylum names and who belongs into each phylum. And then, so the bryophytes do not have true leaves, stems, roots, right? That's important. But then you compare that to the seedless vascular plants. Now you do have true leaves, roots, stems, and you have vascular tissue. But they both need water for fertilization. They both release spores. So kind of think about similarities and differences between those 2 groups. Wednesday, when we're together Wednesday, I can answer any, I'll leave a little time at the end of lecture for more questions you might have. And then Friday is the exam.